Welcome all to our June edition of the TB Think Tank webinar series. The session is titled Progress in the Development of Host Multi-Biomarker-Based Tests for the Diagnosis of TB. I'll now hand over to Dr. Karen Dupree to introduce today's presenter. Over to you.
Thank you, Nowazi, and welcome everybody. I have the privilege today to introduce Novel Chegou, who is a professor in the Division of Immunology at Stellenbosch University. He heads the TB Diagnostic and Biomarker Research Laboratory within Stellenbosch University's Immunology Research Group. And his work mainly focuses on the discovery of biomarkers for TB, including biomarkers for the diagnosis of TB and for the monitoring the response to TB treatment. And his current work he currently works on studies that are focused specifically on the diagnosis of TB in both adults and children, which is why I'm also very excited to introduce him in my role as task team of the Child Adolescent Maternal Working Group, or task team. And he's focusing also on biomarkers for both pulmonary and extrapulmonary TB, which is really exciting. He's also passionate about translation of biomarker findings into tools that may be used for TB control and as point of care tests. Novel, very welcome, and we're very much looking forward to your presentation and hearing more of your work.
Thank you so much, Karen. And good afternoon, everyone. So happy, grateful for the opportunity to talk to you about some of our work. Am I audible?
Yes, you are, Prof.
Okay, great. Thank you. So I'm going to, after a brief background, I'm just going to walk you through some of the work that we've been doing towards identifying or coming up with a test that can be used for diagnosing pulmonary TB. And after that, I'll go to work that we did on TB meningitis in children, and I'll end up with some concluding remarks. Well, being a TB audience, I don't have to remind you talk so much about limitations of the tests that we know that we currently used for diagnosing TB. But I just want to point you to this survey that was reported in 2017, where the authors looked at the availability of different tests that we're using for diagnosing TB, and to gauge how people who were involved in that space whether their readiness for adopting new tests. What was striking in this paper is that smear microscopy, which we know has so many limitations, was still being used in over 90% of the world's TB labs. And even more concerning is the fact that serological assays, which have been banned by the WHO a few years ago, were still being used in over 25% of the labs that were surveyed. So you ask yourself, why do people use these assays? The main reason is just because they are simple, they're easy to perform, and they give you results immediately, so no waiting time. And so I don't know, for some reason people sacrifice, although they know that they are not accurate, they still continue to use them. So most often when we talk about TB diagnosis, the challenges that we have, most often we're talking about pulmonary TB. We know that extrapulmonary TB is even more challenging in terms of diagnosis. And one common limitation that has been talked about a lot in many papers is the fact that our currently most useful tests perform best on sputum. Okay, so the main issue with this is that most of these tests they will not be useful in people who are not able to provide good quality sputum samples. And here we're talking about people who have extrapulmonary TB, children who find it difficult to expectorate. And so the challenge is coming up with new tools that will be easier to use, and also to perform in samples that can be collected from all patient groups. Recognizing this need, the WHO published this World High Priority Target Product Profiles a few years ago, and this document lists the tests that were identified as high priority if we are to get a hand on the control of TB. And so the 4 key priority tests that were identified are it's a non-sputum based test that be capable of diagnosing TB in all forms, diagnosing all forms of TB. A point of care test which should be simple, low cost, and which can be used by first contact healthcare providers to identify those who may need further testing. A sputum-based smear replacement test, and the other one was a rapid drug susceptibility test. But prior to the publication of these criteria, though, we had already been involved in work aiming at identifying new biomarkers, new non-sputum-based tests for TB. I mean, more than 10 years before that document was published. And how did we start with this work? This is work that started following our observations in interferon gamma release assays, that this test, the new test that were introduced to us then and were being hyped as the solution for TB care or diagnosis, were not really useful in our high burden settings. To summarize, in these studies, we carried out some studies in adults, children, HIV infected, HIV uninfected individuals, and we realized that these tests were not very useful in discriminating between people who were latently infected and those who had active disease. And so at that time, we started wondering, what do we need to do? What would we need to do if we had to develop a new test for diagnosing, especially active TB? And so, given the nice platform that the interferon gamma assays were built on, we started wondering whether we could identify new host biomarkers other than interferon gamma, which was being used in the then IGRAS, or new antigens other than the antigens that were used in these assays. And so we carried out a series of studies where we tried to do just that. So in a study that was done as an add-on project to a much larger study that was being done across a lot of African sites called GC674, we had access to over 100 antigens, which were screening in whole blood assays, like shown here, as to see if they were useful in first, as whether they were immunogenic, and then secondly, whether they were useful as would we have any potential as diagnostic tests for TB. At the same time, we started looking at multiple host biomarkers using multiplex immunoassay platforms that were identified in supernatants that we're harvesting from this test to see whether we could identify any host biomarkers that may be better replacements, for example, for interferon gamma. And so what we're hoping to achieve then was to come up with biosignatures that would combine new antigens and new host biomarkers, which we could take forward for development of simpler tests for TB. In this, one of the projects that we did is that we were involved in. It was a study that was being done in 7 sites, secreted in 6 African countries, called the African European Tuberculosis Consortium. In this project, we developed a first prototype of a lateral flow assay based on up-converting phosphor imaging technology, which we showed to work across the sites that we were working in. And although we thought that the assay was at least promising, we soon realized when we were looking carefully at the data that they were generating that a lot of the biomarkers, that if you feed the data into a statistical analysis software and you tell the data, give me the best combinations between the biomarkers, we noticed that a lot of those biomarkers that were coming up were being detected in the unstimulated samples, which we included in those culture experiments as just negative control. Just mind you, the negative control, if you look at the example of the quantiferon test, the negative control is this gray tube that we collect blood and put inside, and no antigen. It's basically plasma that you leave in the incubator for the duration of the time that you're incubating your assay. And so after making that observation, we started wondering whether we could detect these biomarkers directly in plasma or serum samples. And so we went ahead and designed a small pilot study in which we recruited a few study participants from Ethiopia and South Africa and tried to look at the levels of some of the biomarkers that we were interested in serum samples from these study participants. We were happy to see that some of the biomarkers, that in combination with some of the biomarkers, showed potential in diagnosing active TB. And after that observation, we went forward and designed a larger study, which we carried out in 5 other African sites, field sites. And in this study, we enrolled over 700 study participants who were presenting with symptoms requiring investigation for TB. And after working the patients out and separating them into TB or not TB groups, we then look at the accuracy of these biomarkers in discriminating between the two groups. And in this work, we actually showed, I think then for the first time, that without any antigen stimulation, you were able to come up with a combination between 7 different biomarkers, which showed strong potential for diagnosing TB, as seen with a sensitivity of almost 95%, specificity of about 73%, and with a good negative predicting value. So after this work, we have been doing a lot of other studies as part of other consortia, and a lot of students involved in validating the signatures, trying to optimize the models. And as part of those studies, we reduced this 7-marker signature to a 3-marker model, and also translated the assay to a finger prick-based test. It's a much simpler assay now, where you just collect the blood, prick the blood of a finger, prick the finger of a study participant, collect blood, dilute the buffer, and pour it on the test card like this, leave it to dry, then you measure. So where are we with this assay? So the test was recently evaluated as part of 2 large studies, one of which was funded by the STP, and the other one, really complementary studies, was funded by the NIH across a number of sites. And this is work that was led by Professor Gerard Falser, Professor John Bilal of the Colorado State University, and Jen Sutherland from the Gambia. A manuscript on this work was recently published, actually in just 2 months ago. And yeah, the paper is there. You can download it. Anybody who's interested can download the paper and see how the assay performs. But just to summarize, we enrolled over 1,200 study participants in that study from 4 different sites, and we performed 2 index tests. One was our protein test called MBT, and the other was the Cepheid's expert host response test, which makes use of, which looks at 3 host genes. Using a composite reference standard that took into consideration results of culture, expert, and other measures, we separated the participants into TB and not TB, into TB and not TB groups, and then then look at how the assays performed in diagnosing TB. Well, quickly for our test, which is the MBT, we were able to diagnose TB in this study participant with a sensitivity of 91%, specificity of 73%, and a negative, and importantly, negative predictive value of 96.4%. And how about the expert host response assay? It also performed equally well, sensitivity approaching 93%, slightly lower specificity, but also nice negative predictive value. What we see that was common among the 2 assays is that there was some variability, as you can see in the panel B figures. There was some variability in test performance across different sites. But yeah, like I said, the paper is published. You can have a look. There's a lot of detail in that paper. So is this all that we have been doing? Now, we have been using similar approaches, trying to look at different biomarkers in alternative sample types like saliva, urine, pleural effusion. So we've done this in the context of TB and also various extrapulmonary TB types. But one particular extrapulmonary form of TB that I'm going to focus on, which I've worked on the most, is TB meningitis in children. Okay, I think we have questions at the end. I would have asked for any questions now, but let's continue. Excuse me. So why TB meningitis? Why TB meningitis? Well, it's the most severe form of TB. Mostly occurs in children, young children, and adults who are HIV infected. And the terrible thing about this disease is that 50% of people who are diagnosed with it die or suffer from long-term neurological issues if they are treated. And the main issue with this disease, this form of TB, is not the absence of chemotherapy, but diagnostic delay. And why diagnostic delay? Because symptoms are not specific. Microbiological confirmation of the disease is very difficult. And to diagnose the disease, you have to do lots of investigations on the CSF, and most of the tests that you do individually are not very useful. And so you need neuroimaging, some kind of imaging to confirm the disease. And because of the challenges in diagnosing the disease, Children soon that are referred to hospitals like Tigerberg Hospital, where we are based, have been to the clinic up to 6 times, up to the hospital up to 6 times before eventual diagnosis of the disease. And this is data that is similar. This is data that is not unique to our scenario here. It's the same thing. You speak to people in Vietnam, they will tell you that exactly the same thing, an average of 4 visits before the disease is diagnosed. So a few years ago, we started wondering what are some of the biomarkers that we're working with had potential in diagnosing TB meningitis. So we performed this first study where we look at the evaluation, where we look at the expression of some of the biomarkers that we were working with at that time in children with and children without TB meningitis. And we came up with that. At that time, we came up with a 3-marker signature containing these proteins, which showed some potential in diagnosing TB meningitis. You may think that this is not too sensitivity of 52% and 95% specificity. You may think that it's not too great, but really there are no simple tests that perform this much. So after having seen these results, we secured some funding, some seed funding from the Technology Innovation Agency, recruited a few more study participants, and then tried to validate the signature that we identified. In that study, we observed that if we substitute 2 of the proteins, we placed 2 of the proteins that we're working with new proteins, we could improve the accuracy of the CSF-based test. At the same time, we also identified a 3-protein signature that was detectable in blood, which also showed potential as a diagnostic tool for childhood TBM. Yeah, as part of an ITSP senior fellowship that I've been running for a few years now, we've done lots of validation studies of these signatures. And in one of those studies, where we were looking at the CSF signature, we observed that the 3-marker, 3-protein signature that we have been working with and an alternative signature that we have also been looking at continue to perform well in new study participants. Additionally, in this particular study that was published in 2022, we could come up with new combinations of proteins, which showed even slightly improved accuracy. But because we have been validating this signature over the years, we know that it seems to work. We went further and tried to convert it to an ELISA-based platform to see how it performs in the form of an ELISA. So in this first kind of proof of concept ELISA study, which we did in just 28 TB patients and 28 children with TB and 46 without TBM, we showed that we're able to reproduce the results that we're using, whether we're generating with the Luminex, more expensive Luminex platform. And when we look at the proteins together, we could also reproduce the results that we have been getting. And in fact, our ELISA data show that we may only need 2 protein biomarkers in a test, and not all the 3 that we're working with. So how far have we gone since then? Well, I think 1 or 2 years ago, we started collaborating with a Cape Town-based assay development partner called Life ACDX, and through a project that was funded by the South African MRC through the MEDIC platform. And in this project, we're trying to convert that signature, 3-marker signature that we've been validating, into a lateral flow test. So 5 months, just under 5 months into the project, Louis, whom you see in this picture, showed up in my office with these boxes like this, with prototype tests that they have developed for us to have a first initial look at, a first evaluation at. If you look at the test, you see it's quite simple, similar to any other lateral flow assay that healthcare workers will be used to. Actually, in fact, even people, this is similar to the home pregnancy test that some of us who are married, do have done a few times. And what we did at that time was just the initial evaluation, technical evaluation was focused on just seeing whether the assay, just looking at the technical aspects like testing different sample volumes, looking at different read times, etc. And after that, we gave a report to the company for them to use to further optimize the test. As I was traveling back from a meeting that I went to last week, ITSP Forum last week, on Friday, I heard that the company delivered the final optimized test to the lab. So after this presentation, I'm going to be meeting with the team, and we're going to plan the evaluation of the final versions of this test in stored samples to see how it will have a first idea about the clinical performance. So there's other work that we are still doing, that is ongoing on the subject of TB meningitis in children. And this includes validation of biomarkers that we identified previously in blood. And these include both protein and mRNA-based signatures that we have been working with. And we are doing this in samples that we collected from Tigerberg Hospital. And at the same time, we've secured some samples from African and non-African partners, and the samples are being shipped to us as I speak. And then just to put what I've been saying in context, I want to just look back at the benchmarks that the WHO set as the target product profiles for new non-sputum based test. And what did they say about a community-based test? Well, they are saying that an ideal test should have a sensitivity of 95% and a specificity of 80% against microbiologically confirmed TB. But the acceptable targets, which is what all test manufacturers are aiming for really, is a sensitivity of 90% and a sensitivity of 70%. For extrapulmonary TB, with the adults as the benchmark, it was difficult to get data. So there was no lower range of sensitivity that was specified, but they advised that a sensitivity of about 80% for CSF, if you're using CSF, I guess TB meningitis would be acceptable. How do our tests look? Before even getting to how our tests looked, there has been a lot of talk about these target product profiles. Many people question whether the target profiles are realistic, are they too high, are the targets too high? But a modeling study was published last year, and in this study, these authors look at the trade-offs between the clinical performance of a test versus how simple the test is to perform. And what they basically concluded in their modeling exercise is that diagnostic tests that bring results closer to the patient could reduce overall diagnostic loss. And so what they are saying in this study is that if you have a test that is easy to perform and the results are accessible near the point where patients are enrolled, then a sensitivity ranging from 70% should be sufficient for the test to make an impact. Now, how do our tests look? Well, if you look at the pulmonary TB test, we meet the benchmarks that the WHO set. And from what I showed you, if you may recall, it should be a finger prick based test should be very accessible if it is based on it's a simple platform, such as the lateral flow that I showed you. And for the extrapulmonary TB test, the TBM based test that we are developing for children, that one also exceeds the requirements for that were set up by the WHO, even against the adult benchmarks. So to conclude, what I've been trying to tell you is that we have developed at least a prototype finger prick-based assay that may be useful for the diagnosis of TB at points of first contact with healthcare providers. So point of first contact with patients, and we're thinking about community clinics and day hospitals. We've also developed a CSF-based test that may be useful for diagnosing TB meningitis in children. And both of these tests meet the WHO's target profile requirements for their intended use cases. And if you look at the modeling paper that I referred to now, the fact that these tests are very simple, should be simple to perform, means that, yeah, if implemented carefully, if used appropriately, maybe they may make a good contribution to the control of TB. There's a lot of other work that is going on, like I mentioned, trying to optimize, especially the blood-based signatures, so that we can come up with maybe blood versions of the test. And we continue to work with industrial partners. We have quite a few students, including engineering students, who are looking at new point of care prototype formats. And at the moment, we're also currently talking with our tech transfer office in Novus about the way forward with the assays that we've developed so far. We've been also talking to a few stakeholders just to gauge their opinions and insights into what we could do from here. So to end, there's a lot of people that have contributed to especially the two major studies that I've shown you, and I cannot include all of their names on slides, but I'd like to single out a few individuals. Professor Gerard Walson, with whom I've been working on this. We've been working on this since the beginning, 2005. Professor Riga Solomons, with whom we've done all the TBM work, pediatric TBM work with. Professor Fanny Marhaba and the BMRI clinical team, which has been instrumental in the adult pulmonary TB work. Charles Maniello, who was a previous PhD student who is now completing his MD degree. Sister Conisa Claassen, James Sidon, and other colleagues at the Department of Pediatrics and Child Health that we work with on the TBM studies, our lab team, and the consortia that we've been using to evaluate the pulmonary TB tests. It's a lot of people in the ATP consortium, the screen TB, triage index, and then the TBM kits. And then, yeah, my lab, we've been very fortunate to train quite a few smart students. Some of them contributed significantly to this work. They have left, and there are other ones who are working on the projects as I speak. And yeah, they've been contributed. Their contribution cannot be ignored. And finally, our asset development partner, Life SADX, represented by Louis Rowe, and then the funders. Without the funding, none of this would be possible. Once again, thank you very much for your attention. And I look forward to any questions.
Thank you so much, Prof, for the presentation. Thank you. Thank you. I see the applause from the people in attendance. Thank you so much. We have a question here. Our first question asks, what price do you expect the finger stick test to cost, or do you have a price target?
Thank you for that question. So at the moment, we have the honest truth is that we are busy porting because the finger stick test we developed it with academic partners. We are now looking at the possibility of porting it to another platform that is in collaboration with the asset development partner that I just mentioned. And from all indications, what I've heard is that it should cost less than $2. Should cost because it's quite low cost material that they're using in those assays.
Okay, thank you so much. Dr. Mamoto, would you like to speak to your question? Are you able to unmute and speak to your question?
Yes, good afternoon, colleagues. Thanks for the comprehensive presentation, and thanks, Nolwazi. I just wanted to know, how long does the CSF lateral flow assay take to read? I think we know like with the urine test, it takes about 25 minutes, and we know some of the associated complexities when implementation happens on the ground. But I just wanted to find out, how long does this one take? Thanks.
Thank you very much for that question. So, like I mentioned during the presentation, we did the initial pilot testing, a couple of samples of the test that the Life Assay gave to us. And what we did, we tested times ranging from 10 minutes to 30 minutes. And we think between 15 and 20 minutes is the optimal time that will work for that assay. So we are going to, for the new evaluations that we're going to be doing on it now, the final version of the test that we delivered last week, we're going to, we still, like I said, I'm going to be meeting with the team now so that we really look at the SOP, but I think it's likely going to be 20 minutes.
Thank you so much, Prof. Prin, are you able to speak to your question as well?
Sure, just a point of clarification, Prof. Are all the evaluations that you've done only on children, or do they also include adults?
You mean for the TB meningitis test, right?
Yeah, and for the finger stick.
Okay, the finger stick is only adults. Yeah, it's only adults. We've done some work where we look at serum samples in children for pulmonary TB. Yeah, so that one is mostly adults. But for the TBM test, we've done some work in a few adult samples, but it's not many. Majority evaluations that we've done has been in children. So, and how does the signature perform in adults? We have not really tested CSF samples from adults because these were really the clinical group team that I work with is pediatricians. So it's pediatric neurology. So we focus on children, but we have access to some adult samples, but these were all serum samples. The biomarkers that we work with, that we identified for children in serum samples, unfortunately, is not the same. They don't work well in adults. So it looks like if we're going with a blood-based test, then it will be a different test for adults and children for the context of TB meningitis.
Are you covered, Bren?
Yeah, and just to clarify, the finger stick, then you haven't done any evaluations in children, and I assume you'd also get a different kind of profile of response.
Yeah, so in children, it's a bit tricky. So we've been involved in a number of studies. We were not the PRs of that study, in collaboration with colleagues at Imperial College. So a study that they recruited study participants from across Africa, also in Cape Town. And there are some common targets. There are common targets between the adult test that we have developed and the signature that we have there. So yeah, there are some common targets, but it's not exactly the same. I don't know whether that's okay. You want me to elaborate more?
Thank you, Prof. Thank you, Pren. Next up, we have Graham. Graham as well. Prof Menkes, are you able to speak to a question as well?
Yeah, can you hear me?
Yes, we can.
Yeah, thanks, Novel. Really exciting to hear about the progress of your work, and I know how much has gone on in the background to develop these findings. Just one question, if you can expand on the assessment of the 3-protein diagnostic in blood for pulmonary TB in adults. That study, the setting that it was done in, was it primary care among patients presenting with respiratory symptoms? So were those that were negative for TB people with other conditions that mimic TB, and so therefore reflecting how it would be used in a real world clinical setting?
Thanks for that question, Graham. Yeah. Actually, yeah, that's a very important question. Yes, actually, yes. So the assay was done in primary healthcare settings. So what we did was we went to typical the health centers that we've been working in, and patients that came in, people that came in, and who were what's the right word now? Suspected. Don't know what I suspected. Presumed to have TB, who were being investigated for TB. We recruited all of those people, and then we performed the test, and then performed all the reference standard tests, separated them into TB and non-TB groups before doing the assessments. So it's actually patients that came to presented at primary healthcare levels and with symptoms requiring investigations for TB. So as in that study, I did not show that figure, but in that study, we also did profiling of the people who were diagnosed with in whom TB was ruled, and we profiled them, and we look at a viral panel, see whether they had alternative bacterial infections or viral infections, and we observed that some of the tests were not really effect of especially viral, those co-infections, including COVID-19, were not really, did not affect the test much. For the MBT though, there was a slight effect if a patient had a bacterial infection in the other respiratory disease group. There was a slight effect, but not really significant.
Okay. And then just a follow-on question. I saw that you categorized some participants in that study as possible TB. And so there was definite, probable, possible, and not TB. How did you handle how big was that group of possible TB, and how did you handle that in the analyses?
So possible TB, if I recall, well, what is that? Yeah, if I recall, possible TB was was just 5 individuals.
Okay. Yeah. So very
just
small.
5 individuals that had possible TB. So with this, it's difficult. You cannot. We just excluded them from the analysis.
Yeah. No, so it wouldn't have affected it too much. Yeah. Thanks very much, Novel.
Okay, thank you. But we also look at the ability of the test to classify both the expert host response and the MBT to classify the possible and the possible TB group and people who we Yeah, and people who were not classifiable using the conference post algorithm. And I think the test assigned one of these people, diagnosed one of them as, was it one or two of them as TB? Said one of them could be TB and the other one's not TB. So we try to look at how the test perform in predicting those groups as well.
Thanks.
Thank you. You're welcome.
Thanks, Prof. Menkes. Mohlopeni, are you able to unmute and ask your question?
Yes, I am. Thank you.
Thank you.
Novel, beautiful work, and congrats on your beautiful, results. So my question is almost related to grams in the context of whether it's possible to incorporate TB-specific antigens in finger sticks to rule out any other diseases that may present with inflammatory markers. Generalized, because if actually you don't have a TB specificity there, one would actually think of that it could be another underlying thing. So, but it's just about the utility, the possibility of having an antigen incorporated in a finger stick sort of lateral flow device.
That's a very good question. Yeah, and honestly, it's something that we've thought about. Some of the studies that we've done, we've looked at different panels of TB antigens in serology, the serological assays. There are some antigens that seem to work, okay, especially when you use them in combination with cytokines. We publish some work like that. It will be interesting. It will be interesting. It's just that then it makes the assay a little bit challenging to develop. But I agree, it's something that we should consider doing. Especially we think about that also for the assays that are based on samples collected from the site of disease, like the CSF. If we can include a TB antigen in that kind of test, then certainly, if the TB the biomarker that the TB specific protein shows positive, then it's TB. So it's a little bit challenging, but I think it's something that is doable. It also depends on the threshold, how much of that antigen can you detect in blood compared to the host biomarkers that you're trying to look at. So yeah, it's something that it's doable, but yeah, we surely look at that as time goes on. But thanks, very good question.
Thanks, Novel.
Thanks, Prof. Novel. Nogolunga, are you able to unmute and ask a question?
Thank you, Nolwazi. I wanted to ask if this is intended to be a facility or community-based test. And I wanted to know if this could be used as a screening test.
Okay, so thank you for that question, Nolwazi. Oh, sorry, I did not get your name correctly. So yeah, it is. We intend to use it as a triage test, especially the finger stick assay. This one that is shown here. So the plan, what we are thinking will happen is that if you are sitting at a day hospital, for example, and you have this long queue of people that are in front of you, and people come complain about their symptoms, then instead of just collecting samples and saying, oh, collecting samples, say go and collect sputum samples, sending for gene expert, then waiting for results. If you have suspect that person may have TB, you perform the test right there and then you get the result, and it would let you know whether you can then do a confirmatory test such as the gene expert. So that's the intended use case. So it's supposed to be used as a triage test to identify patients that may need further testing. So what the benefit that you can get from what we're hoping that the benefit that this test would give is that instead of testing 100 people to get about 30 TB people with the gene expert, they're waiting for the results to come back. If you're, for example, if you're in a setting where in Cape Town where your sample needs to go to Greenpoint, then you have to wait for maybe 2 weeks for the gene expert result to come back. You do a test then, and it tells you that the person is positive. If a person is positive, then you only do a gene expert on that one. So that's the intended use case. And if the test is negative with a good negative predictive value, you can say, confidently say the person likely doesn't have TB. But for the TBM test, though, the application may be different because that is, at the moment, we've only worked on participants that were referred to Tigerberg Hospital. These are people that have been to the hospital over and over, like I said, up to an average of 6 times. So the use case is a little different there.
Thank you so much, Prof. Do we have any other questions, comments for Prof Novel?
Prof, just to pick up on that point, do you not envisage use as a screening test in a community setting, where you, for example, are systematically testing particular high-risk populations?
But so if I can get it clear, as part of active case finding study, is that what you mean?
Yes, yes.
Yes. Okay, thank you. So so we only reporting what we've done, right? At the moment, we've only looked at people with symptomatic TB, people who come to the clinic, people whose symptoms have been gone to an extent where they think I need help, then they go to the hospital. So for people who may have TB, but who do not yet feel sick enough to go to seek healthcare, we're doing some work on that at the moment, but we're still looking, we have some data that we're generating on those kind of study participants, but I can't really say whether I will work or not at the moment in that setup.
Thank you. Thank you, Prof.
But that would be a very good use case as well. If it works, then that would be great.
Thanks, Prof. Nobulunga, you may go ahead.
Thanks, Nolwazi. So I'm actually interested. I was excited, Prof, when I saw the ease with which this test would be conducted, anticipating that this is a test that is intended to be used at the community, or rather to be used within the community level. I think I'm concerned that with all the work that's going into this, we are yet again targeting interventions for case finding, centering them in healthcare centers instead of at the community where we really should. I mean, data from, I don't know, countless previous studies has directed us to the community for case finding efforts. Why are we still creating, rather triaging efforts to be intended for facility use? Over.
Thank you so much, Nupulunga. That's a very good question again. So, like I said, we only report what we've done, right? This is the data that I showed you is from, patients that we enrolled in facilities. Like I said, I have a student who just had his PhD in March this year, and whose work was based on looking at some of these targets in that kind of patient group that you are talking about in the communities. So there is some potential, but we're still doing this is work like this. What I'm showing you now is work that we've done over many years. We've done many validation studies before coming up with this. So it's still early stages. So I cannot really comment to say whether this is going to work or not, but it may work. This test may work, but we need to actually prove that we need to get the data. We need to get the data to say whether it really works on really. So thank you. That's
Thank
a
you,
really good
Nobulunga.
question.
Thanks for the questions. Perhaps, Prof. Novel, I see the colleagues that you worked with are on this call as well. Do you think perhaps they'd like to chip in or assist perhaps with closing remarks or any comment from them?
I can see. Yeah, yes, please. Okay, now I can see. Yes, please, colleagues, if you want to chip in, say more, please just expand on something.
And can they just raise their hand so I can quickly unmute them? Whilst we wait for the colleagues to raise their hands, I just would like to remind everyone on the call that if you'd like to see yourself on this platform in the coming months, please do email myself at nkosi@tbthinktank.org if you'd like to see yourself presenting on this platform in the coming months. Okay, we have a hand. You may go ahead, Dr. Chendi. You may unmute.
Oh, hi everyone. And thanks. Oh, can you hear me?
Yes, we can.
Oh, okay. Yeah, so it was quite an interesting talk, and thanks, Prof, for it's always interesting to listen to all of the talks again. And to me, it's someone who have also worked on this biomarkers while looking at the performance of the signatures in the non-African side. It's quite interesting to see where the work is going and the usefulness of these biomarkers. So yeah, it's quite exciting.
Thank you. Thank you.
Thank you for your comment. Any last takers for any questions and comments before we end the session a bit early today? Any comments, questions? If somehow you do have any questions once you've dropped the call, you're also free to email myself and I can put you in contact with our presenter today, Prof Novel Chegou. That is also possible. With that said, I think I will reward everyone here with an extra 13 minutes for them to have their lunch or stretch their legs. And we are getting thank you messages. Thank you again, Prof Novel Chegou, for the amazing presentation. Very insightful. And thanks again for everyone on the call for your questions and comments. And thanks again for your time.
Thank you so much, Uluwazi. Thank you, Tim.
Thank you, everyone. Enjoy your day further. Thank you.